Dear learners, welcome back to NPTEL online certification course on novel drug delivery system. I am Dr. Satish Devanapelli, Assistant Professor at Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. In our novel drug delivery system course, we started with the various types of the novel drug delivery systems. In our last lectures, we have focused on the various types of the gastro-retentive drug delivery systems. Today, we are going to discuss on the new topic, that is microencapsulation. So, this is the today's flow of presentation. First, I will discuss on the what are the microencapsulations and how we will prepare various types of the microencapsulations, and then we will focus on the structures, types and composition of the microencapsulation systems, and further we will discuss the factors that will affect the microencapsulation during the synthesis, and then we will focus on some of the mechanisms which involve in the drug release from the microcapsules. And after that we will focus on the various methods of the microencapsulation, then we will focus on evaluation and the applications. So, microencapsulation is a novel technique. Here we use this system as a novel drug delivery platform to convert various types of the conventional molecules and conventional products into the drug delivery systems. Here this technique we use to encapsulate our active ingredient by surrounding with some of the materials which are also called as the surrounding materials that will protect the molecules and that will help in the control the drug release at the different pH conditions of the body. So, here the definition is says that microencapsulation is the process of surrounding or enveloping one substance, that is active ingredient, which will come in the core within another substance that is called as a shell. So, in the picture you can see this is the core, and here we load our active ingredients. Sometimes it may be single or it may be more than one type of the active ingredients we can load into this system. And after that we will use various types of the coatings based on the number of active ingredients. We can coat with the various types of the polymers. So, that is called as shell, and yes, the size is very small, and which we will get in a capsules. The size range from 1 micron to 1000 micrometer. So, the product which obtained by this process is called as microcapsules. So, here we can see in the pictorial representation. So, here we will get in a capsules which are in the range of the micron size. So, here it is also known as the protective technology. Here we use various types of the active ingredients either in solid forms and the liquid material. Some of the cases we can also use various types of the volatile ingredients which will encapsulate and will show their performance completely differently from the conventional formulations. So, here we encapsulate solids and liquid materials. Upon that we will coat with the various materials which are the coating materials, and then you will get in a micro particles, and this field is utilized in the various applications. So, pharmaceutical, chemical, cosmetics and the food and the textiles, we can also use these formulations in various routes. So, here this technology is widely used in the various types of the domains. So, why we need to prepare microencapsulations when compared with the conventional formulations? As we know that in pharma there are the drugs which suffers with the lot of physicochemical problems and also in the therapeutical problems, bioavailability issue and the solubility issue. To overcome that, microencapsulation method is one of the attracting platform. With that, we can encapsulate various types of the drugs which undergo the degradation. For example, vitamins and the peptides which will degrade in various biological and the at room temperature conditions. In such cases, we can use such sensitive molecules and that molecules we need to encapsulate in the core material. So, with that we can avoid the degradation of the sensitive molecules by using microencapsulation. And as we discussed, yes, we can convert liquids into the solids because liquids are unstable in various conditions. In such cases, if you would like to convert your liquids into the solid dosage forms, the microencapsulation is the best strategy. We can overcome all the handling and the storage issues. And not only that, we know that most of the antibiotics and other drugs which are bitter in taste, to overcome that we can also mask the taste by using polymers as a microencapsulation technique. And then yes, if you would like to target at some particular site by using pH sensitive polymers and temperature sensitive polymers, you can definitely use your active ingredients and by using of these polymer materials as a coating materials that will definitely reach into the particular site and there your drug will be released. So, which is called as targeted delivery. With that you can avoid the side effects by using the microencapsulation. And not only that, when you would like to go with various drugs which are incompatible with the excipients or two drugs are incompatible at particular conditions, such cases we can separate by coating with the polymers and after that we can add another drug. So, we can with that we can avoid the incompatibility problem with the drug components and excipients in a single dosage form. So, that is also one of the attractive strategy in the pharmaceutical industry. And here you can see the core material, that is the inside of the carrier, and here you can see the wall material, that is also called as the coating materials, which may be various types of the polymers, waxes and the resins which will work as a wall material. And here you will see there are the stability issues that may be because of the temperature and the oxidation and the pressure that will affect your molecule and that will degrade various sensitive molecules. So, with this we can use as a protective agent for the sensitive molecules. We can also use as a targeted and also yes, definitely this polymer will show the control release compared with the conventional systems. So, the objective of the microencapsulation technique is to maintain the sustained delayed release when compared with the conventional formulations because of the polymers that will undergo the swelling and also they will follow various types of the release mechanisms. With that we can control the release in the microencapsulation format. And yes, we can provide accurate dosing because you by end of the day you will understand how much drug is loaded per capsule or per amount of some capsules. So, with that yes, as a unit dosage forms in case of tablets we can say that we can deliver the accurate dosing. Similarly, here also with this also we can provide the accurate dosing by understanding the amount of the drug which are presented in the microcapsules, and especially that is which are the drugs, the potent and the low dose, and that will affect the patient. So, such cases we can use it. Already we have discussed this point, which we can protect from the various degradation strategies. So, with that we can also avoid the degradations, and once you follow all these, definitely we can improve the bioavailability, therapeutic efficacy, and we can also reduce the side effects of the active ingredients by using of the microencapsulation techniques. So, here now we will see the structure of microencapsulation systems. So, here you will see here, as we discussed in our previous novel drug delivery systems and also in the introductory classes of the novel drug delivery systems, there we have discussed the microcapsules, also the microspheres. So, now here you can see the capsules are nothing but which contain two systems within one dose. Here which contains two different systems, that is, one is core and another one is the shell. So, shell is made up of the polymers, resins and waxes. Core contains the active ingredients. So, we can separate these two things, whereas matrix or microspheres, here the drug is embedded in the polymers. So, here it is a matrix systems. So, here you will not see any two different layers, or we cannot separate the content from the polymers. So, whereas in core and shell, if you want to protect your molecule by using the shell, it is possible. So, both release will be different when compared with the conventional systems. So, here the structures you can make core and shell type of the microencapsulations, and also you can prepare matrix based microencapsules. Yes, the drug distribution here in case of core and shell, the drug is available on the core and shell will help for the protection, and both cases we can expect the sustained controlled release and flexibility also possible, because we can use various types of the sensitive molecules and the size ranges from the 1 to 1000 micrometer. Here we can use like taste masking, volatile drugs and also liquids also we can convert into solid dosage forms. So, these are the structures of the microencapsulation systems. And here we will see the types of microencapsulated systems. So, based on the various methods of preparation techniques, so we can prepare various types of the microencapsulated systems. So, here in the picture you can see, you will understand what are the various types of the microencapsules. So, here the first example is mononuclear, that is single core. So, here you can see it contains one single core which contain active ingredients. The outer layer is the shell. So, one core surrounded by the one shell, commonly used for the sustained release. So, in case of multi-shell, here you can see the core is one and there are so many shells are there. You can see it looks like a onion like structure. So, multiple shell layers around the core offers extended release and the stronger protection. So, whenever you would like to go for extended release and for the stronger protection of your active molecule, such cases multi-shell system is preferable. That means you can say layer by layer techniques. And also the another topics, that is the polynuclear or multi-core. So, here this is quite opposite to this system, because here you can see this is a one core and here you can see the various types of the n number of core is available, and here you will see the one shell which is surrounded by the multiple core systems. multiple small cores enclosed in a single shell gives better uniformity in the release. So, with this we can also get a uniformity in the release. And as we discussed just now on the matrix type, so here the drug, that core is embedded in uniformly in the polymer, or you can say the wall material. So, yes, so that is dispersed in a wall material, and yes, there is no distinct shell. And whereas irregulars, so here you will see there is no proper shape of the core as well as shell. Asymmetrical or non-uniform shapes useful for the burst release whenever you want to go for the immediate release, and such cases irregular systems are feasible. And then we will see the composition of the micro encapsulated systems. So far we are discussing on the what are the micro encapsulations and what are the microcapsules, and we have seen the structures and then types. Now we are going to see what are the materials which involve for the preparation of the microcapsules. So here the first one comes, that is a core material. It may be active ingredient which comes in the solids and the liquids, like various ingredients like pharma grade or food grade, nutraceutical, cosmeceutical active ingredients, all this can come into this category as a core material. So, this is our active molecule, and there are second platform is nothing but coating material. So, here you can see the list coating material which we coat or we will surround the active molecule on the surface of the active molecule. So, here the polymers, waxes, resins, proteins and the polysaccharides will involve as a coating materials. And third one is the vehicles, that may be aqueous and non-aqueous. So, this will be selected based on the solubility of your core material as well as coating material. So, it will again depend on the solubility, physico-chemical properties of your core material as well as coating material. Sometimes this may vary because of your method of microencapsulation. When you want to go with the non-aqueous, such cases we need to prefer various types of the solvent other than the water. And if a drug is water soluble, then you can prefer aqueous. When you would like to avoid water, if a drug is undergo like hydrolysis, and such cases we can go with the non-aqueous systems. Now, here is the examples of various coating materials. It is classified based on the synthetic and the natural materials. So, here you can see again it is classified on the based on the non-degradability and the biodegradable polymers. This all we have discussed in the first week, that is on the introduction to novel drug delivery system. There we have discussed on the polymers and the usage of the polymers in novel drug delivery systems. So, yes, we can classify synthetic polymers based on the non-biodegradable and the biodegradable polymers. And then here you can see the natural materials. Yes, this also we have discussed the what are the advantages of the natural and the synthetic polymers, and these are the examples like albumin, gelatin, collagen and the carbohydrate classification, and there are the chemically modified carbohydrates, nothing but semi-synthetic type of the materials. And also we can classify based on the chemistry. So, various types of the biomaterials or you can say the coating materials are available. And another class comes, that is the resins and then waxes and then enteric resins, which we will use for the enteric coating. And again in the resin we have water soluble and the insoluble. Whenever you would like to go with the insoluble, or you can say if you want to go the long sustained release, such cases the insoluble resins are preferable, which will help for the control release. And here these are the few factors will affect the microencapsulation. So, you can see these are the various factors and this will impact on the drug delivery. For example, consider what is the role of core material in the microencapsulation. So, here yes, that will affect your stability, solubility and the loading efficiency, because this will decide the final outcome of your microencapsules. So, that will affect your stability, solubility and the loading efficiency. Whenever you face any issues, you need to modify your core materials. So, such cases we can improve the various types of the microcapsules. So, wall materials also very important, that will affect your the release steady, encapsulation efficiency and also protection. The concentration, type of wall material will affect the outcome of final dosage form. Particle size also will affect, as we discussed, the size should be 1 to 1000 micrometer. So, the lesser number of the size, the uniformity also will important, and that will also affect the control release and the tissue distribution. So, the less number is preferable compared with the higher number of the particle size. Method of encapsulation, yes, that is also one of the major role that will definitely influence the your uniformity of drug content in the polymer, and also the cost also will help, and also the scalability. So, always method of encapsulation should be preferred the economical and the scalable technology like spray drying and other type of the methods. Release mechanism, that will depends on the type of material, and ultimately that will affect the final release of the core material in the polymers. pH sensitivity, that will also help in the, as we discussed in case of the entry coating polymers. So, definitely with this also you can expect the target drug release. Stability, that is important factor, that will help the shelf life and the performance under various environmental stress conditions. So, here these are the some of the drug release patterns. These are already we discussed in our previous lectures of the gastro-retentive drug delivery system, and also we discussed same parameters in control release drug delivery systems. So, here this will follow diffusion control, erosion control and the swelling, burst release, and you can also, it will depend on the pH and enzyme trigger release. So, such mechanism will depend on the type of the wall material. So, for this entire presentation, for this entire slide, you can prefer our control release drug delivery. There we discussed various mechanisms of drug release. So, here these are the various methods of microencapsulations. So, with this we can make by using of the core materials and the wall materials, we can use various types of the various methods of the microencapsulation techniques. With that we can encapsulate the bioactive molecules, which we use for the not only for the foods, also in the pharma and the cosmeceuticals and nutraceuticals. So, the first technique is spray drying. Here what we do, will homogenize our drug and polymer, and then will atomize into the hot air. Then our solvent will evaporate, either it may be water or ethanol, methanol or IPA, isopropyl alcohol. So, then you will get the dry particles. So, here you can see the core material and your wall material, this need to be dispersed into these and processed through homogenizer, and you will get an emulsion, and that need to be spray into the chamber, and then you will get in a dried powder. And yes, this method is very easy to operate, flexible, economic and the fast, scalable compared to any type of techniques. And yes, here you cannot use heat sensitive molecules, that is one of the limitation, and after this you will get the completely dried product. Co-observation technique, this is also one of the most commonly used technique after the spray drying. So, here what we do, we will precipitate our wall material with the active ingredient by altering the pH, the temperature and the addition of the electrolytes and the non-solvent systems. By these 4 principles, we can make a co-observation techniques. So, there you will see the precipitated wall materials, and is one of the most commonly used technique in the industries also, and it will give the high encapsulation efficiency, and this technique will help in the control release, and this have a wider application in the food technology also. So, here this co-observation can be divided or classified into 2 systems, that is, first one is the simple co-observation system and another one, second is the complex co-observation systems. So, here the phase separation is facilitated by promoting the interaction between the macromolecules at the cost of macromolecule solvent interaction with the help of the electrolytes or by the addition of the solvents. So, here sodium sulphate and desolvates we use as an electrolytes, and we use solvents, that is ethanol, or manipulating the temperature. So, we can make a simple co-observation techniques. So, the important point is that here we use only one polymer in the simple co-observation, whereas in complex co-observation we use oppositely charged hydrocolloids. For example, here we can use sodium alginate and we use calcium chloride. So, both are opposite in charge, then it will make a complex. So, multiple polymers we use, especially by using electrostatic interaction. So, plus and minus, these 2 polymers will make a complexation, and with this we can make the complex co-observation microcapsules. Now, here this is various steps involved in the complex co-observation. So, here we have 3 things. One is your core material, and then you have a vehicle, and you have a polymer. You have the that is wall material, that is polymer. So, we need to disperse this system, and when you add the opponent system, so it will start form the separation. We will see the separation of the 3 things, and then you will see the once you increase the concentration of one of the material. For example, if you are increasing the concentration of wall material or by increasing the electrolytes or solvent systems. So, with that what happen, you will see initially you will see the separation, that is you can see formation of immiscible phases, and after increasing this concentration, so then it starts forming the core material, forming the coating on the core material, and if you further increase the concentration of the opponent material, so it will undergo the rigidization. So, this can be classified into 3 things. One is formation of immiscible phases, deposition, and the rigidization of the coating materials. So, ionic gelation is very simple technique. Here also we use the 2 opponent systems like ionic crosslink system. Here also we use calcium alginate bleeds. You can see here sodium alginate, and here is a calcium chloride that need to be transferred into the calcium chloride solution, and here you will get the microemulsion that is not in the dried form. After that we can add another opponent charge, that is a chitosan, then you will get a beautiful microspheres after the drying. Here you can see the microcapsule formation, rinsing with the water and then coating with the chitosan, and then we can remove the microcapsules. So, here the we can use various types of the proteins and peptides for the microencapsulation. So, these are the another techniques which are upcoming and recent in the microencapsulation methods. Here we use the SNADs, self nano emulsifying drug delivery systems. So, here also when you would like to improve the solubility of the drugs, here we can use lipids and surfactant. By using some energy based technique, we can improve the solubility. Electrospinning and electrospraying is also one of the attractive thing. Here we can use heat sensitive molecules. So, two different polymers we can conjugate or complex, and then we can do the microencapsulation. This is preferable for the probiotics and proteins. And supercritical fluid also, this is one of the great technique. With that, this CO2 will work as a solvent, and this is a non-toxic in nature. With that, we can produce microcapsules at low temperature. This is a very clean process and green technology, and the tunable size we can make various types of the shapes also. So, this will give a efficient coating in the microcapsules. Now, these are the few evaluation studies. So, here we can evaluate the particle size by using optical microscopy and laser diffraction, that is master sizer. With this, particle size is very important, that will affect the control release and also stability and encapsulation efficiency, that can be reported in the micrometer. By using scanning electron microscopy, we will understand the surface texture, shape and porosity. This will give the morphology of your microcapsules, whether it is smooth or rough, that will also we can find out. So, encapsulation efficiency, with this we will understand the amount of the drug presented in your material, or you can say the final microcapsule, that can be quantified by using UV or HPLC method. So, here drug content, what is the drug content meaning? That it will give the amount of the drug per unit microspheres. So, this can also be performed by the various spectrophotometric methods, UV and HPLC. This is very important to evaluate for the microcapsules. In vitro drug release, that is based on the pH. We need to select right pH medium, and we need to perform the dissolution studies, and we can use various simulated conditions, and again further we need to quantify by using spectrometric and HPLC methods. So, after that we can use release kinetics. With that we understand whether your microcapsule following diffusion, erosion and swelling also. So, and also there are the stability studies, percentage yield, swelling index, and there are other studies also you can perform as an evaluation techniques in the microcapsules. And these are the few applications. In the pharmaceutical industry, we can use a microencapsulation technique as a control release, taste masking and also for the target drug delivery. In the food, we can use various flavors, vitamins and the probiotics, other food materials. We can use it for the encapsulations. Cosmetics, we can use various actives in the creams, we can convert into the microcapsules that will sustain the release. In agriculture, we can use pesticides and other nutrients that we can deliver into the crops. And in biotechnology, if you want to encapsulate various types of enzymes, peptides and other genetic material in the biotech industry, the microencapsulation is one of the attracting and emerging platform for encapsulation of the various active ingredients. So far we have discussed on the what are the microencapsulation, then we have discussed the structure, types, and also we have seen the various methods of the microencapsulation techniques, and also we have seen the various types of the applications, and also we have discussed the various types of the microencapsulation techniques. So with this we are going to stop today's lecture. In our upcoming classes, we are going to discuss on the other types of the novel drug delivery systems. Thank you so much.